Hello friends, again welcome to my channel. Today we are discussing about one of the important and interesting experiment which is carried out within the genetics, that is the Meselson and Stahl experiment. Now majorly we know that the DNA which is present as an genetic material within all living organisms except some type of the viruses. The Watson and Crick in 1953 first put forward the structure or the model of the DNA, and along with that, majorly they explain that the DNA must be present in the double helix form, means it is an double stranded helical structure. It is an right-handed helical structure. The nucleotide bases form the interior sugar phosphate backbone, means the four type of the nitrogen bases which are observed within the DNA, that is adenine, guanine, cytosine and thymine. They are combined with the deoxyribose sugar and the phosphate group, and combinedly they are responsible for forming the monomeric units of the DNA, that is the deoxyribonucleotides, and preferentially there is the generation of the DNA molecule. According to the Chargaff's rules, there is the presence of the same number of purine and pyrimidine bases within the DNA. Now all of this, it is about some information about the structure of the DNA molecule. Along with that, the Watson and Crick also explain that majorly the DNA molecules, these are get replicated through the semi-conservative mode of DNA replication. Now, no doubt they explain that as the DNA it must be replicated, means as the DNA it acts as a genetic material, so it has the ability to get replicated to form its exact copies. It is called as the replication. So they put forward the model for the DNA replication as the DNA must be replicated by the semi-conservative mode. Now this semi-conservative mode of DNA replication, it is get explained by the Meselson and Stahl, that is the Matthew Meselson and the Franklin Stahl. Both of these scientists, they perform an experiment to prove that majorly the DNA it must be replicated through the semi-conservative mode of DNA replication. Now there are the three different types of the major hypotheses. These are put forward by the Meselson and Stahl. Now the first one, it is the semi-conservative hypothesis. According to this semi-conservative hypothesis, if the DNA it must be replicated by the semi-conservative mode, then from the one parental DNA, whenever there is the synthesis of two DNA molecules or the daughter DNA molecules, within that, firstly there is the separation of the two strands of the DNA, and each of the parental DNA strand it acts as an template strand on which there is the synthesis of its complementary strand. That means within the synthesized 2 DNA molecule, one of the strand it is completely the parental strand, and another strand it is the newly synthesized strand. It is called as the semi-conservative mode of DNA replication. Now the second one, it is the conservative mode of DNA replication. According to the conservative mode of DNA replication, there is the copying of the intact DNA double helix, means there is no separation of the DNA molecule. Directly the complete DNA molecule it acts as the template strand, and whenever there is the synthesis of the two DNA strand, one of the DNA molecule it is completely the parental strand on which there is the synthesis of the new DNA molecule. Now the third type of the mode of the DNA replication or the hypothesis, it is the dispersive mode. According to the dispersive mode, majorly there is the fragmentation of the parental DNA strand, means it is get converted within the different fragment, it is get dispersed within the different segments, and all of these segments these are get copied, and whenever there is the synthesis of the two DNA molecule, there is the mixing of both of these molecules, that is the old strand and the new strand. Now we are switch over to the Meselson and Stahl experiment. Now this Meselson and Stahl experiment, it is carried out in 1958 to explain the semi-conservative mode of DNA replication. For that purpose, majorly there is the utilization of the two different isotopes of the nitrogen. As we all know that the nitrogen, it is the major component present within the DNA. That is, as there is the presence of the nitrogen bases, the main component present within this, it is the nitrogen. Means for the synthesis of the DNA, there is the requirement of the nitrogen within the growth media. So whenever there is the growth of the Escherichia coli cells, within the media there is the presence of the ammonium chloride, that is the NH4Cl. As within that NH4Cl, the nitrogen which is present, for that purpose there is the utilization of two different nitrogen isotopes. Now these nitrogen isotopes, these are having the different atomic masses. No doubt the N15, it is having the heavier mass than that of the N14, and the N14, it is lighter one, and it is commonly observed within the cells. And majorly after the rounds of the DNA replication, it is observed that within the synthesized DNA molecule, which type of the nitrogen isotope was observed, on that basis it is explained that either the DNA replication, it is carried out by the conservative, dispersive, or by the semi-conservative mode. Majorly within this experiment, there is the utilization of these two different isotopes of the nitrogen for the purpose to label the DNA molecules, that is to label or to differentiate in between the parental DNA and the daughter DNA molecules. Means for the purpose to differentiate in between the parental DNA which contains the N14 and within the daughter DNA which contains the N15. Majorly for that purpose there is the labeling of these DNA molecules by using these two different isotopes of the nitrogen. Now the N14, which is the lighter one, it is commonly observed within the cell, and the N15, it is rare one, which is the heavier element. Majorly now for differentiation in between this heavier and the lighter nitrogen isotopes, or the DNA containing this either the heavier or the lighter isotopes, there is the utilization of specific centrifugation technique which is called as the density gradient centrifugation. Now this density gradient centrifugation, within this there is the utilization of the cesium chloride. This cesium chloride, it is responsible to differentiate in between or to separate out the two chemical compounds which are having the different atomic masses, or on the basis of their densities they are responsible to separate out these molecules. So for the separation of these DNA molecules, that is either with the lighter isotopes or the heavier isotope, there is the utilization of this technique. All of this technique, it is utilized to detect whether the new nitrogen atoms appear on the first or both of the daughter strands. Now we are switch over to the methodology for the Meselson and Stahl experiment, which is carried out within 1958. So for the performance of this Meselson and Stahl experiment, majorly they grew the 14th generation of E. coli in the N15, that is the heavier nitrogen isotope. Means majorly the E. coli cells which are utilized as the host cells for studying this mode of DNA replication. Majorly these E. coli cells which are initially containing the N14, means within their DNA there is the presence of N14. Such type of the E. coli cells, these are transferred to a growth medium with the NH4Cl which contain the N15 isotope. N15 isotope means the first step within this, it is the N14 containing E. coli. These are grow within the media where there is the presence of N15 isotope, and about the 14th generation of these E. coli cells, these are grown within the media containing the N15 isotope. Now after such type of the 14 generation, again these E. coli cells which are grow within the N15 media, again these are get transferred to the second type of the growth medium which contains the N14 instead of the N15, and again the generations of the DNA replications these are carried out. Now within the different periods or within the gaps of these, majorly periodically the DNA sampled from these E. coli growing E. coli cells, these are extracted and majorly these are utilized for the purpose of density gradient centrifugation to compare the percentage of both of this nitrogen isotope, means within the DNA molecule at which amount there is presence of the N14 and the N15 isotope of the nitrogen which are acts as the main components within the DNA molecules. Now for this density gradient centrifugation, this DNA sample extracted from the E. coli cells, these are mixed with the cesium chloride. Then there is the centrifugation, it is carried out at the higher speed, and then these are allowed to settle down for the purpose of formation of the bands on the basis of their densities. Now how many types of the result they may be obtained within this? Now if suppose we are considering about, as we say that the N14 containing E. coli cells or the N14 containing DNA which is present within the E. coli cells, these are transferred to the N15 media. These are transferred to the N15 media, and then the first round of the DNA replication it will occur. We know that the E. coli cells, these are having the 20 minutes of generation time, means within the 20 minutes there is the formation of two cells. There is formation of two cells, means the DNA replication it is carried out within the 20 minutes. The first round of the DNA replication it is completed within the 20 minutes. So whenever there is the completion of the first generation of the DNA replication, at that time if there is the extraction of the DNA and we perform the centrifugation, at that time they get the result that majorly they are getting an intermediate band, means it is not the heavier and it is not the lighter one, it is an intermediate, band. That means it contains both N15 and the N14, Now again whenever it is get introduced, means if now within this experiment, whenever up to the 14 generations there is the growth of these cells within the N15 containing isotope, at that time they got only the one of the band, that is the band of the heavier DNA, that is which contain the N15, Now if again when they transferred these E. coli cells which are grown within the N15 containing media, these are transferred to the N14, then they got such type of the intermediates. Then again after one generation there are the half of the intermediates, means the half amount of the DNA they are containing the intermediate, and they are again getting one of the another band which is of the lighter DNA, that which only contains the N14, means both types of the bands these are obtained. Means if there is the presence of only N15, then we are getting only the single band. The first replication band located at the midpoint between N15 and the N14, and the second generation had the two bands, that is the first at the midpoint and the another at the N14, and the same type of the two bands these are observed subsequently. Now all of these results which are obtained, that is the two bands which are obtained, that is one it is of the intermediate and another it is of the lighter one, these can be only appeared if the DNA it must be replicated by the semi-conservative mode, because if there is the replication of the DNA it is carried out within the conservative mode, then there is only the appearance of the single band of N15. There is only appearance of the single band of N15 and the N14, because within the conservative mode or according to the conservative mode, whenever there is the DNA replication, one it is the only completely the parental DNA and another it is the newly synthesized DNA. So there is the, if there is appearance of both of these strand, the N15 and the N14, then and then only it is the conservative mode. But here within this experiment they obtain the intermediate band, they obtain the intermediate band. Now if suppose it is an dispersive mode, then within the dispersive mode no doubt there is also appearance of the intermediate, means these results these are similar within the semi-conservative mode and also within the dispersive mode. But if again it is an dispersive mode, there is only appearance of the intermediate band which is present, which is the mixture of both N14 and N15, But again whenever the subsequent generations these are carried out within the N14, it is observed that the another band of the lighter nitrogen, lighter nitrogen, that is the N14, it is also observed, which is not possible within the dispersive mode. So from all of these discussions it is concluded that majorly the DNA it must be replicated, by the semi-conservative mode, which is get proved from this Meselson and Stahl experiment. Now within this figure you can easily observe that is within the first generation there is the presence of only the N15. Okay, means here only the single type of the nitrogen it is present. Now whenever the media it is get changed to the N14, then there is the only presence of the intermediate band. There is presence of the intermediate band which contain both N14 and the N15, means in the density it is in between the N14 and N15, it is called as the intermediate. Okay, now after the third generation, again there is no doubt there is the presence of the intermediate, and along with that intermediate again it is also containing the another band which contains the N14, that is the lighter band. Again after the fourth generation the same condition it is observed. So all of this result it prove that the DNA it must be replicated by the semi-conservative mode, within which the double stranded DNA double helix at the time of replication get separated, and each of the single stranded DNA from this parental strand they acts as a template strand, and on which there is the synthesis of the new strand, and then this parental DNA it combines with the newly synthesized strand and form the DNA double helix. Means within the synthesized and newly synthesized DNA molecules, one strand it is completely the parental strand or the old strand, and the another strand it is the newly synthesized strand. Such type of the DNA replication which is considered to be the semi-conservative mode of DNA replication. So it is the all over information about the semi-conservative mode of DNA replication or the Meselson and Stahl experiment. So if you like this video, please subscribe my channel and also share this video with your friends. Thank you.